again. Imagine seeing you here. Today we are going to look at the slower, more specific portion of the immune system, the adaptive immune response. As its name suggests, rather than being an inborn set of defenses like the innate immune response, the adaptive immune response develops over time as it adapts to defend against pathogens in which it comes in contact over the lifespan. Let's learn more. First, recall that we need the adaptive immune response because while the innate immune response is good at slowing pathogen growth, it often fails to completely stop growth or completely annihilate the pathogen. Thus, the need for the adaptive immune response, which can finish off what the innate immune response started. However, the adaptive immune response has super high specificity. Its response is to specific antigens unique to each pathogen, and it is versatile enough to respond to nearly any pathogen. Antigens are small chemical groups usually associated with pathogens that are recognized by B and T cells. When first exposed to a pathogen, the symptoms from the resulting disease are usually fairly severe, as it takes time for the adaptive immune response to be generated. This is called the primary adaptive response. However, subsequent exposures to the same pathogen result in stronger, faster adaptive immune responses because they have already seen and won this battle before. Symptoms of subsequent infections are usually mild, if they appear at all. Many times, the pathogens are defeated before they can even cause an illness. Brilliant! This ability to recognize and respond to pathogens it has seen before is known as immunological memory and is the reason why developing a healthy immune system early in life leads to less illness later in life. Finally, an important benefit of the adaptive immune response is the ability to distinguish between self-antigens and foreign antigens. As B and T cells mature, they learn to ignore self-antigens and only mount a defense against foreign antigens. However, if this self-recognition fails, a class of diseases known as autoimmune diseases can result. Recall that T cells and B cells are the main lymphocytes or immune cells of the adaptive immune response. Of the two, T cells are particularly important as they can exert direct control over the immune response as well as some control over their B cell counterparts. T cells recognize antigens using a two-chain receptor protein, of which the most common is the alpha-beta T cell receptor shown here. Each of the two chains have two domains, the variable region domain and the constant region domain. The constant region domain is closest to the cell membrane and has little variation in its amino acid sequence, hence its name, constant region domain. Its amino acid sequence is fairly, constant. The variable region domain is further from the cell membrane and, you guessed it, its amino acid sequence is variable. This is the part that forms the antigen binding site of the receptor, and it is this variability in the amino acid sequence of both the alpha and beta chain that allows T cells to recognize a wide variety of pathogens. Yay for diversity! We've thrown around the term antigen a few times now, but we should probably discuss them further as they are the key to recognizing foreign invaders and pathogens. Antigens are usually large, complex structures with many antigenic determinants. Also known as epitopes, these antigenic determinants are small regions to which a receptor can bind. In this case, we are interested in them as the binding sites for T cells as shown here. Warning! Important concept forthcoming. You must know that T cells do not recognize the antigens of the pathogens themselves. That's right, T cells can't recognize pathogens alone. So what do? Enter the role of antigen-presenting cells, like the cell shown here. These cells internalize pathogen antigens and break them down into smaller pieces in a process known as antigen processing. They then regurgitate these antigen fragments to the cell surface alongside a special antigen-presenting protein known as an MHC, or major histocompatibility complex. This process is known as antigen presentation. The T cells bind to the presented antigens and become activated. And now for our anatomical analogy. To what shall I compare the antigen-presenting cell? Is it a mama lion? A mama fish? No! An antigen-presenting cell is a mama bird, eating, swallowing, and regurgitating smaller pieces of antigens to present to their baby birds. T cells. Open wide. You should know that antigens from different pathogens use different MHCs. Class 1 MHCs are typical of viruses, while Class 2 are associated with bacteria, parasites, and fungi. Since viruses can infect nearly every body tissue, many cells express Class 1 molecules, but only immune cells express Class 2. The cells of the immune system that express Class 2 MHC molecules are nicknamed professional antigen-presenting cells and include macrophages, dendritic cells, and B cells. Once a mature T cell binds to a processed antigen on the antigen-presenting cell, it becomes activated and begins to rapidly divide using mitosis. This speedy proliferation is known as clonal expansion and boosts the strength of the immune response to a level sufficient to control the pathogen. Because T cells have variable regions, only the ones with the correct binding site will bind to the antigen. This is known as clonal selection and contributes to the high specificity of the adaptive immune response. So, clonal expansion increases the strength of the response, while clonal selection increases the specificity. Together, they are building you an army for disease defense. Let's take a look at a few different types of T cells making up your disease defensive line. Helper T cells secrete cytokines to enhance other immune system responses. Cytotoxic T cells kill target cells through apoptosis, similar to natural killer cells. Regulatory T cells are also known as suppressor T cells. There is much that is unknown about these cells, but they suppress other T cell immune responses, as their name suggests. They have the ability to shut down or regulate the response. During the primary adaptive immune response, or the first exposure to a disease-causing pathogen, the body will generate memory and effector T cells. Memory cells remember the pathogen and are ready to stage a rapid response to future exposures. They can even last a lifetime inside of us. The rapid secondary response results in the production of a large number of effector T cells, which overwhelms the pathogen before it can cause a second round of the same disease. This is the entire basis of immunity to a particular disease, like chickenpox. Once you get it, you are immune to subsequent infections. From T cells and antigens, we will now move to a brief discussion about B cells and antibodies. Antibodies are glycoproteins. They have two types of polypeptide chains, heavy and light, to which carbohydrates are attached. These molecules have two identical heavy chains and two identical light chains, making a characteristic four-chain structure. Of particular importance is the Fc region, which is formed by the two heavy chains. Effector cells usually have Fc receptors and can bind to this Fc region. Two identical antigen binding sites are found opposite the Fc region. You might have heard the terms antibody and immunoglobulin. What's the difference? Basically nothing. They are one and the same and can be used interchangeably for the purposes of this course. They are basically a form of secreted B cell receptor that can cause agglutination or clumping of bacteria, thus making them easier to eliminate. Spiffy. There are 5 main classes of antibodies or immunoglobulins: IgM, IgD, IgG, IgA, and IgE. In general, antibodies have one of two functions: to serve as the B cell antigen receptor, or to be secreted, circulate, and bind directly to a pathogen. Of the 5 classes of antibodies, only IgD and IgM can serve in the first capacity and can act as the antigen receptor for naive B cells. Okay, so IgD antibodies serve as B cell antigen receptors. IgM antibodies are responsible for primary responses, while IgG follows with secondary responses. IgA antibodies are secreted in mucus, tears, and saliva to kill pathogens, and IgE antibodies are involved in allergic reactions and parasitic infections. So what exactly happens with B cells the first time you are exposed to a pathogen versus future exposures? As you can see here, there is a delay in antibody production after the initial exposure. This primary response can have an initial delay of a few days as the B cells clone and take time to expand and differentiate and mount their defense. During this time, the antibody supply is low but sufficient. Subsequent exposures do not have this delay and result in an immediate secondary response with rapid production of B cells ready to fight the infection. These antibody levels are much higher than the primary response and can easily overwhelm the pathogen to prevent symptoms from even developing in the first place. Yay! Finally, let's close with a look at active versus passive immunity. Active immunity is called active because it requires the immune response to take action. It is an adaptive immune response due to the direct exposure to a pathogen. This direct exposure can occur naturally through daily life or intentionally via the introduction of weakened or killed strains of the pathogen through vaccines. Either way, it results in the body mounting a defense and developing antibodies. Passive immunity does not involve direct exposure to a pathogen. Rather, the person receives a direct transfer of antibodies to help defend them from the disease. Yep, they don't even have to make them. Free antibodies. These antibodies can come in the form of immunoglobulins injected into a patient or naturally during pregnancy from mom to fetus. While the whole free ride thing might sound appealing, passive immunity does not offer immunological memory, leaving you defenseless against future exposures. In other words, the protection only lasts as long as you are getting the antibodies. Well, that does it for lymphatic and immune system adaptive immunity. See you next time.